Good morning, everyone. Thanks, everyone, for coming this morning and not being out in the sunshine, I guess, on the first kind of plus degree day in a while. I'm really excited to have Romina Mizrahi here giving our talk. So Romina is a new addition to the Douglas Research Center and McGill Psychiatry as well. And we're very happy to have her. So thanks. So it's been since the summer, I think, summer of 2020 that Romina has been with us at the Douglas. So Dr. Mizrahi obtained her MD degree from the University of Buenos Aires in 1998 and her PhD in 2007 from University of Toronto. Her work seeks to understand the early molecular changes in the brain of youth with psychosis and addictions, with the hope of identifying novel targets for prevention, intervention, and treatment. Dr. Mizrahi has received numerous funds from CIHR, the NIMH, and the Brain Behavior Research Foundation since becoming an independent investigator in 2007. Her work hopes to advance our understanding of how the brain functions in vivo, using, I'm sure you'll see, a lot of PET-related work, and we've had the pleasure of working with her on integrating, along with Lisa Guma, integrating structural MRI with some of these PET markers in really interesting samples. So, I'm excited to see how it all comes together in today's talk. With that, I'll let you take it away, Romina. Thank you.
Thank you, Malara, for the nice introduction. Thank you, everyone, for being here today. A little bit of a disclaimer. I have given this talk at ACNP in December 2020. I'm a little bit sleepy today for some reason. I can't wake up, so I will see how this goes. I'm going to do my best. You see, I will be talking about molecular imaging studies in youth, and I do this on purpose because based on the interest of the audience, I can change the presentation that I give with this title, which has been very useful over the years. So I'm going to be starting anytime you would like to ask me a question. I'm happy for Malar or anyone else to interrupt me. There's no problem. The most important thing is that the message gets translated. Some conflict of interest and disclosures. I have received some support and speaking fees from Janssen. In terms of the C4A TSPO work, which is the unpublished part of the presentation today, I want to thank Marco Sanchez, biostatistician from CAMH, Bob Sipursky and Ann Bassett, respectively from the University of Toronto, who have given me insightful comments, and two students from Toronto, Sina Hafizi and Tania da Silva. And I realized I should have added here Elisa Guma. I was thinking about this last night, but I haven't. But whereas Sina and Tania have worked around these 3 studies or 4 studies I'm going to be presenting today, Elisa was key for the analysis of the C4A TSPO work. So her acknowledgments should go up here. This work was supported by NIMH funding to look at neuroinflammation in psychosis and psychosis risk. So today I'm going to be discussing the neuroinflammation or TSPO set of studies that we have initiated in 2013. I'm going to walk you through the process of translating a new radioligand all the way from the bench to the bedside. We're going to be discussing F18 FEPPA, which is a 2nd generation TSPO radioligand. Imaging TSPO, then we are going to be looking at imaging TSPO as a marker of neuroinflammation in the psychosis spectrum. We're going to be then discussing neuroinflammation in cannabis use disorder. And finally, we're going to be integrating this with recent analysis looking at brain complement component C4A, as well as TSPO and brain structure. Many of you are familiar with this clinical characterization of the clinical high-risk state for psychosis. Basically, this is a state before a major psychotic break. And this is people who start to have some functional deterioration, some early type symptoms, and they come to us with emerging symptomatology. About 1/3 of them end up developing a full psychotic break. And we have clinical ways to identify them using structural interviews such as the SIPs or the CARMs. And as I've said, 30% of them will convert to a full psychotic break within 2 years. This is so much difference from the characterization of MCI for Alzheimer's disease. About 2/3 will not convert to psychosis, with 1/3 continuing to have ongoing symptoms, long-term functional trouble, and 1/3 recovering 100%. So obviously, we need ways to identify who is going to go on each path, as well as, of course, for psychosis in general, schizophrenia specifically, we are aiming to uncover new biomarkers for identification and stratification. So one of those targets is looking at neuroinflammation. In PET, we have used new radioligand, F18 FEPPA. Microglia activation can be quantified with brain translocator protein, or TSPO. There are several caveats for this. When we started back in 2013, there were less. As time passed, we learned a lot about this target, but nonetheless, we have been able to do a lot of studies and learn, I think, something about the brain and TSPO. So in its resting state, microglia performs an immunosurveillance role. However, when microglia is activated, it increases the expression of TSPO. This is accompanied by an increase in inflammatory mediators in brain, as well as some neurotoxic substances which have to be controlled to avoid, in theory, long-term trouble for the brain. So increased TSPO expression in brain, as measured with PET, is a reliable marker of neuroinflammation. Although, as I've said, it's not without caveats. So FEPPA is was a novel TSPO radioligand. The prototypical TSPO radioligand up until that time was carbon-11 PK11195. However, it has several limitations, which prompted the development of the second generation radioligands, such as DAA1106, which was mostly used in Hopkins, and PBR28, and DPI713, and FEPPA, which was developed in Toronto by Alan Wilson. PBR28 is the one that's used, developed by NIMH and used widely across the world. F18 FEPPA is an F18 version of this carbon-11 PBR28 radioligand. So it allows for more clinical use, given that has a longer half-life with an F18 as compared to carbon-11. So the first step, of course, was to establish the radiation dose and establish that it was consistent with other F18 radio-level ligands, which we did back in 2013. And then Dr. Pablo Rostron was able to model with the two-tissue compartment model using arterial input function to reliably obtain the total volume of distribution, which is our outcome measure for PET. This is a measure that's measured in the whole brain, and it's using kinetic models, basically using blood to estimate the expression or the density of TSPO in brain. Now, at the time, a group in NIMH who were studying PBR28 found that there was a wide variability with PBR28. As you can see here, a binding can go all the way from 5 to 17 or 17. And so they identified a polymorphism in the TSPO gene, RS6971, to affect binding, such that high affinity binders here who have the CC genotype have about 20% binding as compared to the MABs or the medium affinity binders. This is established with blood, looking at this SNP, and there are some people, very few, less than 5%. I think it's 1% in Asian population are labs, so low affinity binder, and all those labs we cannot quantify with FEPPA because we cannot see anything. So everything you're going to be seeing, therefore, it includes only HABs and MABs, and we correct for this RX6971 polymorphism in every single study that I will be presenting to you today. And the same happens with PBR28 studies and any other second generation radioligand for TSPO. The first generation PK1195 does not require to obtain or to correct for this polymorphism, but that's because it has very low sensitivity. So you cannot, or it's not necessary to do this because the sensitivity is too low. So we started by looking at neuroinflammation across aging, and you can see here we scanned a large sample of healthy subjects all the way from 18 years old to almost 90. This is divided like all the studies you're going to be seeing today, controlled by RS6971 polymorphism. And you can see we did not find an association between age and FEPPA, BT, or TSPO. Now, there are some other studies which were done after this study from 2014, which did find some effect of age. I should mention that this group was an extremely healthy cohort, namely these people above 70 to 90. They were people with absolutely no comorbidities and no concurrent medications. So this was a healthy sample of really super healthy individuals, which may explain why we did not find a relationship in our cohort with age and TSPO. The first studies we've carried out then was in Alzheimer's disease. We did this with Alzheimer's because it's a validated neuroinflammation disease as compared to at least schizophrenia at that time. Several other studies were done at that time. Also, this was published early on. I can't remember, I think it was 2014 or 15. Molecular Psychiatry, and we found a significant increase of TSPO density across the brain, which is very significant, about 40% and up. And this was corrected for partial volume effects. We can discuss this in the end. And this was replicated many times in several studies after that. We also did an MCI study, in which we did not find a difference between healthy and MCI patients. But this helped us to establish that indeed TSPO could be used and was sensitive to a disease model of neuroinflammation such as Alzheimer's disease.
Romina, we just have a question. Lonnie, do you want to ask aloud? Or?
Yeah, I don't mind. I was wondering, given the limitations to TSPO, what is like the current state of the art PET technique for assessing neuroinflammation?
Oh, you want the future. Okay. So there are several ways. There are third generations TSPO radioligands, basically a little bit better to quantify TSPO. And that's one option people can take. Alternatively, we can choose to identify a different target for neuroinflammation. And that's what we are aiming to do here in McGill. We'll see, we still have to model and work around quantification of these novel targets, such as P2X7, for instance. It's a purinergic receptor, which helps us, which has been identified to be a good marker of microglia activation as well. But we'll see how that develops over the years.
Thanks.
Yes, any other question?
No, I think we're good.
All right, so we've went all the way from Alan Wilson's synthesizing FEPPA for us. We've done the safety and quantification, genetic analysis, healthy aging, and Alzheimer's. So we were ready to move on and look at this in the psychosis spectrum. Just very briefly, alterations in the immune system have been implicated for decades in the pathophysiology and schizophrenia. And this is just a very small summary, not inclusive, but sufficient to prompt the studies that we started to carry out back in 2014 or 15. So genome-wide association studies have repeatedly implicated the major histocompatibility complex, MHC, over the years, and specifically more recent findings by Sekar et al. In 2016 looking at C4A enrichment. We're going to be discussing this at the end. Several studies have looked at elevated peripheral levels of pro-inflammatory cytokines, prostaglandins, C-reactive proteins, schizophrenia, and in clinical high risk. Anti-inflammatory agents in adjunct with antipsychotic medication can improve positive symptoms, cognitive and global symptoms. Neuroinflammation may be involved in gray matter volume reduction in schizophrenia, and several epidemiological studies show a strong association between prenatal inflammation and increased risk of developing schizophrenia in offspring, consistent with preclinical studies using inflammatory models. this is not exhaustive, and there's certainly negative studies here, but this was enough for us to, and for others, to start this line of work. So the first study looking at TSPO in schizophrenia using the first generation radioligand, PK1195, came in 2008 by Van Verkel. And as you can see, there were several studies with different characteristics, chronic patients, first episode, treated, untreated, that have been published over the years. Today, I'm going to be discussing our contribution to the literature, and that's the King study in 2015, the Hafizi study in 2016, and the Hafizi study in 2017 as well. Okay, you can see the results here, but hopefully you'll stay with me anyways. So we wanted to use FEPPA to examine neuroinflammation in the psychosis spectrum. We wanted to explore the association between neuroinflammation as measured with TSPO, gray matter volume, symptom severity, and neuropsychological deficits. And based on the literature I just described to you at that time, patients in psychosis spectrum will show higher neuroinflammation as compared to healthy volunteers, our outcome measure being F18 FEPPA VT. Now remember, total volume of distribution, or FEPPA VT, it's an index of TSPO density in brain. So this is the first study we did back in Toronto. It included in this study 27 healthy volunteers and 18 patients with schizophrenia. This was a group of chronic patients. What was interesting for me for this study was that these were people who were relatively sick. As you can see here, the total scores for PANSS are 70, but the positive scores was 19. This is important because we, as the first step, we wanted to have patients who were experiencing psychotic symptoms in a relatively higher number or severity, so that if there was anything, we could actually pick it up. So this was the first, and these were all treated patients, as you can see here, mostly with an atypical antipsychotics. Some of them were taking anti-Parkinsonian agents, and the chlorpromazine equivalents were variable because it was a chronic sample, going all the way from 300 milligrams chlorpromazine equivalents all the way to more than 500 or 600 milligrams. And so all the figures you're going to be seeing are similar. On the y-axis, you have F18 FEPPA VT, and on the x-axis, you have different brain regions. We limit the brain regions for power, but the results are consistent across the whole brain. We found no increase in neuroinflammation in schizophrenia patients with ongoing psychotic symptoms as compared to healthies. We controlled for the RS697-1 polymorphism. We corrected for partial volume effects, especially given the fact that this was a relatively older population. And even when we did this, there was no significant difference between the groups. We confirmed this with voxel-wise analyses, and also we report no association with either clinical or neuropsychological or structural measures in an exploratory fashion at that time. So we thought then, okay, we are going to be studying first episode psychosis patient. If there is a neuroinflammatory profile, perhaps altering synaptic density at the early stages of the disease, this would be picked up more clearly in the early stages of the disease as compared to in a more chronic stage of the illness. So to answer that question, we included 20 healthy volunteers which were matched with 19 first episode psychosis patients. As you can see, most of them were HABs. There were some differences in the amount injected, but this did not affect the outcome. Some of them were tobacco users, only 2 were cannabis users, and only 2 also were taking antipsychotic medication. So you can see here, about 17 of these 19 first episode psychosis patients were actually on treatment. Most of them were not taking antipsychotic medication at the time of scanning. And so for those students carrying out these studies, you can imagine how difficult this is. But they've done a wonderful job in doing that. But however, we found no significant difference between groups, as you can see here at the top. again, reminder, FEPPA VT, or which is an index of TSPO expression. And here you have the different brain regions. The LPFC, hippocampus, MPFC, temporal cortex, whole gray matter. And at that time, we were measuring white matter. We can discuss that at some point in the end. We corrected for partial volume effects, and still we found no significant differences with some correlations. I can't remember now either for with some apathy measures or anxiety at that time in that sample. So we said, okay, then perhaps this is happening earlier in the course of the disease. So we went earlier to look at the CHR, the clinical high risk, and we compare them, or we match them with 23 healthy volunteers from our ongoing healthy volunteer cohorts. As you can see here, 7 were tobacco users, 4 were using cannabis at the time of scanning as quantified with urine drug screens, and we can discuss this a little bit more later. 3 only were taking antipsychotic medications in a low dose, and in general, we say low dose when it's below 200 milligrams chlorpromazine equivalent. So even then, for the 3rd time now, we found no significant differences between the group. We corrected for partial volume effects for the sake of enjoying doing analysis. But as you can see, there's really no differences between groups. What was interesting, however, was that despite the fact that we did not find a significant difference in VTs, there was always a slight decrease. As you can see here, here it's a healthy versus FEP. You see, every time there was a numerical decrease, which we observed in all our cohorts. And so it was Simon Cervenka at the Karolinska Institute who took the initiative twice. This is the first study published in Biological Psychiatry in 2018, and it was done again in 2020, I think, pulling all the studies which have been done to date, to that time, both in 2018 and then again in 2020, and pulling all the studies from all PET centers who have looked at TSPO. He was able to identify, we were able to find a significant decrease in TSPO in schizophrenia as compared to healthies across the different sites, PET centers around the world. So this suggests that indeed TSPO seems to be reduced in schizophrenia as compared to healthy volunteers. However, the effect size is small, and this is why it's very difficult to identify in a single site. Following this very exciting line of work, we have always been interested in understanding risk factors for psychosis, and specifically 2 main risk factors have been explored in my lab for many years. One of them is cannabis use, and the other one has been stress. And in this case, we are going to be, I'm going to be presenting the results from looking at cannabis use on F18 FEPPA. And so the reason we did this is because regular cannabis use has been associated with long-term changes in the brain, making it a growing public health concern in youth, particularly given. Sorry, before I start this, I'm thinking, any questions about this before I move into the cannabis studies? Because I talk fast and maybe people want to ask me any questions about the psychosis studies and TSPO?
Nothing yet, but I'm sure, in aggregate, we'll get something towards the end. Thanks, Romina.
Okay. I talk really fast, so that's why I make this, I stop sometimes to see whether people have questions. Okay, so then we move into the cannabis TSPO studies. As I've said, regular cannabis use has been associated with long-term changes in the brain. Cannabinoid signaling has been shown to play a critical role in the modulation of inflammatory responses. However, clinical evidence remains sparse. Preclinical studies have reported anti-inflammatory and immunosuppressant effects of cannabinoids by inhibiting microglial activation, inhibiting release of reactive oxygenic species, decreasing pro-inflammatory cytokine secretion, and increasing anti-inflammatory cytokine release from microglia. And both THC, tetrahydrocannabinol, as well as cannabidiol are being investigated as potential therapeutic agents for several inflammatory and immune diseases. including schizophrenia and putative CHR participants. So we wanted to examine FEPPA as a marker of neuroinflammation or TSPO expression in long-term cannabis users after an overnight abstinence, and to explore the association between TSPO levels and behavioral measures and inflammatory blood biomarkers. And based on the background that I've just described, and the fact that people really have started to use cannabinoids as anti-inflammatory agents on their own, we hypothesized that long-term cannabis users would show lower TSPO levels in brain as compared to non-users. And so this study was relatively recently published and includes 27 healthy volunteers and 24 cannabis users. Of these 24, about 15 met criteria for cannabis use disorder. Most of them were male, and of course we genotyped individuals, and we also measured cannabis use behaviors. And as you can see, again, our hypotheses were wrong. We found higher TSPO levels in long-term cannabis users as compared to controls. Again, a reminder, on the y-axis, we have F18 FEPPA as an index of TSPO expression. On the x-axis, you have different brain regions. This includes DLPFC, MPFC, temporal, ACC, cerebellum, and the total gray matter, as you can see here. And this effect was even more pronounced when looking into cannabis use disorder individuals, with about 25% increase of TSPO in cannabis use disorders as compared to healthy controls. If you recall, this is less than what we've seen and others have seen in Alzheimer's, which is in the range of 40%, but it's a significant higher effect size or bigger effect size as compared to what was reported in schizophrenia and high risk. And this is why we can identify it in a single site PET study.
Excuse me? I was curious, was there any way that you could either like assess, even in like a broad sense, if people were using cannabis that had more THC or CBD? You mentioned at the beginning that both of them are in trials as potentially reducing inflammation. But of course, we see that like in use, there's very different profiles, especially in recent decades.
Yes, we've measured both THC and CBD in blood. Of course, we could not control what they were using. They were using whatever they like to use. But we did measure in blood. This is very complicated to measure. In fact, we are trying to see whether we can develop a way to measure this here at the Douglas in a very high sensitive way. Just to summarize, none of the patients that we've looked at had measurable CBD levels in blood, and some had THC. And we did find, I think it's in the paper, a relationship with the accumulation of THC in blood. And I say accumulation because there was no relationship with the main metabolite, but with the second metabolites from THC. Of course, I should mention our studies, like these correlations I'm going to be presenting just now here. Our studies really, they're not really powered to look at correlations. As you can see, it's always prone to spurious findings. Our studies are powered to test main hypotheses, but your question is well taken. We did explore this, and as I've said, we found only a relationship with THC levels.
I'm also curious, even anecdotally, do you have a sense of where people were acquiring their weed? Like, was it usually through online distributors or somebody they knew, or maybe this is probably too early, but maybe in recent years also from like the SQDC?
Yes, so we asked them that question. People were doing whatever they were doing. Some of them were using their own dealers. Some of them did try to use the distribution centers in Ontario. They were online only. I think we have to do a better job going forward in trying to identify exactly what people are using. But we couldn't identify any patterns, if that's what you are asking.
Thank you.
So we have another question. Laurie's asking, what about smoking versus ingesting cannabis?
Yeah, so we didn't find any effect of smoking cigarettes or tobacco. There is a single study that was published by the group in San Diego who used not full quantification, so it's a little bit hard to interpret. However, in their analyses of tobacco, they report, it's Brady, that's the author, a reduction rather than an increase in TSPO. So we don't believe that the results we are presenting here are confounded by tobacco.
I suspect he's asking, smoked cannabis versus ingested cannabis. Were they all using it the same way, or?
All of them were using the same way. There was one person only who was using edibles only regularly, and she was included in this. There was nothing different for her. We didn't really investigate single person in detail, but we the studies along the years that we've done with different radioligands, there was nothing specifically different about the single person who was just using edibles.
So one more question. What do you think kind of, maybe you'll talk about this in a second, what do you think makes the levels of inflammation go up in these individuals? Like, are they predisposed? Are they, are they treating some underlying condition as cannabis users that they're not aware of, kind of thing? Or do you have any ideas about that?
Yeah, so it's an interesting question, right? So we have absolutely or very little knowledge of what cannabis is doing in the brain. There's some understanding for brain structure, but for neurochemical changes, it's really very little. In general, most of the studies have shown when looking at different, receptor levels, D2 receptors, I think they've done D1. Well, whatever people measure, there's always a reduction in the expression or the activation while people are using cannabis. So this increase was surprising in many ways, but some preclinical studies have shown that indeed chronic use is associated with increase in pro-inflammatory markers, but not acute. These were regular cannabis users, and so this may explain the concurrent long-term use which was related to TSPO expression. Now, we are now actually submitting a study together with Pablo to look at abstinence. So the idea being, we cannot, of course, give people cannabis chronically from start and offer them to do that from young people. But in collaboration also with Rachel Rabin, what we are going to be doing, we are going to be, hopefully, if it gets funded, we are going to be recruiting cannabis user individuals, and we're going to have them abstain and remeasure an overmarker of neuroinflammation. First, to see whether indeed there is an increase when one measures with another microglia target. But secondly, to see whether this normalizes as it has been shown to normalize with other neurochemical markers. So regarding my thoughts, to answer your question, I think it's possible to find an increase in long-term cannabis use due to long-term use. In general, as I've said, everything in cannabis use disorder, chronic use has been associated with a reduction, except as the studies from Marco Layton and others have shown a potential increase during Qs. I don't think TSPO is sensitive enough to look into this, the Qs piece. And it's not necessarily relevant, but what is relevant, and I'm going to get into the talk, is the fact that TSPO has been associated a few times now to be a marker of stress and depression, or to be associated with stress and depression. So in other words, people who have higher levels of chronic stress, as well as people who have mood symptoms, have repeatedly been shown to have neuroinflammatory phenotypes, and similarly to have higher TSPO. So what may be happening is maybe in a subpopulation of cannabis users who are concurrently developing or who have either anxiety or mood symptoms, these are the people we can potentially see an increase in TSPO. We couldn't look into this individually, but it seems to me that TSPO may be elevated both in chronic use, but also when these comorbid behavioral presentations are occurring.
Okay. One last question. We have one from Marco, who's here. So were the cannabis users abstinent for 12 to 24 hours or longer? If so, could the elevation be a rebound effect?
Yeah, no, they were overnight abstinence only in this study. So we asked them to abstain from overnight, and we didn't measure relationship with hours since last use. It's not here, so I'm remember there is nothing specifically between TSPO and last use, and so I think the new study looking at abstinence and comparing overnight abstinence versus protracted abstinence will be able to answer that question better. Okay, so I think I've mentioned this, right? Chronic stress stores are, of course, are associated with the LPFC, but it was across the brain, TSPO levels. And what was interesting, there's been lots of debate as to whether one can measure in blood or estimate in blood TSPO levels in brain. There's lots of debate about this. And in this particular cohort, and we're now exploring this in schizophrenia as well, there was a significant relationship between CRP levels, high sensitivity CRP levels, and TSPO expression. Here it's in the DLPFC is an example, but it's across the brain. And this is a collaboration with Cindy Wickard from Australia and New York, who helped us do these studies. So, as I've said, the studies in psychosis seem to suggest some involvement of mood and anxiety and stress, but not so much for psychosis. And this is why perhaps we found an effect in cannabis use disorders. So because we had a large sample of schizophrenia patients across a big sample of studies looking at TSPO, and there was this newer study from Sekar et al., which had identified C4A expression in brain to be prominent in schizophrenia. And because C4 promotes synapse elimination by microglia in preclinical models, consistent with lower synaptic density in schizophrenia and lower gray matter and spike density in postmortem and imaging studies as well. And also because microglia has a critical role in regulating synaptic pruning and neuroplasticity in the brain, and complement receptors are highly expressed in microglia, we were interested in further testing an association between genetically predicted brain C4A expression and TSPO in brain, as well as brain morphology in a large sample in the psychosis spectrum, including the cannabis users. And also, we wanted to quantify for the first time C4A expression as genetically predicted as a function of the dosage of each four structural elements. Apparently, this is very hard to do for genetists. It took Jim Kennedy about 3 to 4 years to develop the methodology, but you know, he did a wonderful job in helping us quantify C4A expression, which we did based on Sekar 2016 et al. Paper. So now we have 129 individuals for whom we had TSPO PET data. Of those, there were methodological issues. We ended up with a sample of 111, which included 46 healthy volunteers, 43 high risk, and 40 psychosis patients. This is all the samples that we have collected over the years. And of course, therefore, some of them were nicotine users, some of them were cannabis users. All the samples were included in this study. And this is the main outcome of the study. What you see in the y-axis is F18 FEPPA-BT corrected by TSPO polymorphism. And on the x-axis, you have genetically predicted C4A brain expression. And as you can see here, genetically predicted brain C4A expression was significantly associated with TSPO. The main effect was medium to large, and consistent with previous studies, we found a significant effects of sex. and current cannabis use, which is expected based on the previous study I have just shared, whereas males and cannabis users had higher TSPO levels with no clinical group effect, which is also expected given that there was no clinical group effect in the individual studies. And of course, the sample size for PET highly diverges from the sample sizes in genetic studies. We are talking about several orders of magnitude from 100 large PET study to about 10,000 in genetic studies, which explains why we may not find the clinical group differentiations or effects that is found when people do genome-wide association studies. And so Malar and his team was nice enough to help us look at the relationship between C4A expression and hippocampal surface area. And you can see Elisa, who did these studies, found a negative relationship between genetically predicted brain C4A expression and hippocampal surface area. Now, the interesting thing about this, in my view, is that this hippocampal remodeling seems to be located in an area of the hippocampus which has been previously involved in schizophrenia. And so that's interesting. And you can ask more questions to Malar about this. And we then, we report here the first study looking at C4A in clinical high risk and schizophrenia is second paper in Nature. As you can see, we did not find a significant reduction as he has found, but this is obviously, as I've explained, because of the difference in sample size. But in our sample, at least, we found a significant reduction of brain C4A expression in clinical high-risk individuals as compared to healthies. And if you think about this, there was a relationship across the spectrum between C4A and TSPO. You can understand now why we would have found a reduction in CHR of brain C4A, right? Because we found a significant there's a numerical lower TSPO in clinical high risk, which perhaps is because we have subsampled, without wanting to do so, a group of low C4A subgroup from the larger sample. And so in summary, higher genetically predicted brain C4A expression was associated with higher TSPO. Higher genetically predicted brain C4A expression was associated with altered hippocampal morphology, including reduced surface area. This study is also the first to quantify brain C4A expression in CHR. And we also show a robust effect of sex on genetically predicted brain C4A expression, which is a replication from 2 studies from the group at the Broad Institute, and an effect of both sex and cannabis use on TSPO, which is expected based on the cohorts that we have used previously. And with this, I'm happy to take additional questions, acknowledging Jim Kennedy, as I've mentioned before, Elisa, who has done all the structural analysis for us, Sina and Tanya, whom I mentioned at the beginning, and the patients who have been kind enough to do these PET studies. I'm talking about 2-hour PET scanning with an arterial line. So I'm extremely grateful for their efforts. And with that, I want to thank you for your time. Oh, and funding, of course.
Thank you so much, Romina. Fantastic work. We have time for questions. Lena at the front of the line, naturally. Go ahead, Lena.
You know what you mean by naturally, Malar?
Well, I would expect nothing less, is all.
Thanks for inviting me. This is a great talk, Romina. I really enjoyed it. Thank you very much for letting a small number of us from London to join as well. It's very intriguing what's happening with the inflammation story in schizophrenia, and largely due to the body of work that you produced. I think we're getting some clarity here in the brain basis of this. My question is a general question, really. You know, the more I apprise the TSPO literature and the microglial activation profiles, I'm left with the question: what does microglial activity in brain really represent? The reason I'm asking that is, if you look at some of the mice studies, where LPS, immunogenic agent, the lipopolysaccharides were injected. The mice actually show increased microglial activity immediately, but some of them show even after a year after a single LPS injection. So is this a scar that is left after inflammatory activity, or is it continuous active inflammation that we see, or is it even a healing process that we are seeing when we see increased FEPPA uptake? What is your thoughts on this?
This is an excellent question. Two comments. One is we did try to look at this this effect longitudinally. We worked in collaboration with Richard Bassinet, and I think you've seen the meta-analyses on post-mortem data, where there was some discussion about the neuroinflammatory profile of patients' brain. And in that collaboration, we tried to look into the timelines for the resolution of inflammatory profile as well as TSPO changes. And we could not identify a timeline within the studies that we were doing together with Marco, who was the student at that time. So, I mean, I think this is a great question. It's an open question. We don't know the answer to this yet. I think we don't have the tools to quantify this longitudinally, at least not with PET. And of course, doing longitudinal PET is always problematic because of radiation exposure. And so I think I'm going to be trying to answer those questions longitudinally going forward. But again, our ability to do this in a short time frame will be hindered by the fact that we cannot scan people repeatedly within a year with PET. Now, the interesting part for me will come out from a non-human primate study that was carried out in Yale, where they looked at both microglial activation as measured with TSPO as well as synaptic density. And I like that because now there are studies emerging looking at synaptic density. We're going to be measuring synaptic density here as well. And those studies have reported lower synaptic density as measured with SV2A as a marker in schizophrenia associated to healthies. Now, in this non-human primate, what small number, of course, of animals, what they saw in a longitudinal fashion while started antipsychotic medication on that case, there was a concurrent increase in TSPO while there was a reduction in synaptic density as measured with PET, suggesting that maybe there is an active process happening there between microdeactivation and synaptic pruning. There's lots of discussion about this, whether this happens during adolescence, we are catching the right time or not. Certainly we need to do a lot more going forward. But this is, as much as we know today of this, and certainly finding ways to look at this prospectively will be important.
Just to add to that, I think all this longitudinal work is challenging, but several insights are coming from what you've already shown. There is some relationships between state markers such as anxiety and TSPO uptake that you've shown. So I think there has to be some relationship which is time limited with microglial activity, but some of them may be, a scar tissue that's left for a longer period of time. We are trying to combine some fMRI connectivity analysis in our hybrid PET-MR system with the TSPO measures. Very small numbers, we're just starting now. Maybe we'll get some insights about how functional disintegration happens after TSPO uptake is determined in a group of depressed patients. I will keep you posted.
Yes, I would love to hear more. You may have seen, we did try to look into the relationship between TSPO, glutamate, TSPO, and glutathione. If I remember correctly, there are some relationships there which need to be explored further. And so indeed, it may be that the brain is responding to some type of acute presentation, and then there's a scar. Certainly with PET, we cannot answer that question really very well, but understanding the association between PET and other MRI measurements will be helpful, I think, going forward. So I'm looking forward to the results of those.
Thank you very much.
Thanks, Lena. We have another question from Sylvia. I don't know, Sylvia, do you want to ask it yourself, or would you like me to do it?
Yeah, and actually, I would have another question before. So just in line with the discussion that was happening, So was there any study using an anti-inflammatory drug that crossed the blood-brain barrier? I can think, for example, about naproxen. So using TSPO before, doing the drug, and then kind of scanning after to see if we can remove the signal or part of the signal. Have that been done, or?
Yes. So usually in PET, we do that to validate the radioligand, right?
Yeah.
We want to make sure that that whatever we're measuring, it's a specific. And so for TSPO, this happened after the fact because PK1195 was developed before the blocker for TSPO existed.
Yeah.
The study was done by Ilan Rabiner, the group over in London, and they did find that you, with a TSPO blocker, you can completely remove the TSPO signal. So it's a specific, if that's a question. That's
the question, because I mean, that would suggest that then it's kind of not a scar, right? It's kind of really something there that you can remove. So it's not like a scar on the brain. So it's really kind of linking that to the previous question.
Yes. So okay, so I haven't answered on your question. So what I've mentioned before, it was about the validation of the radioligand, and that is done in healthy volunteers. Now, in patients, it was Jeff Mayer in Toronto. I think he just published it. I haven't read the full paper, but he was looking at TSPO before and after minocycline. I think there was a very small effect I can't remember whether he did it. He must have done it in depression, right? Where there is an increase. Otherwise, you can measure you really need the pro-inflammatory profile in the brain to be able to reduce it. Otherwise, you can do it. Unless it's a blocking study with a drug, like the study I just described to you before. So in the study from Jeff Mayer, I'm assuming, I don't remember, he rescanned a patient with depression who had been previously reported by himself and others to have high TSPO. And in his study with aminocycline, I think there was a reduction, but it wasn't a massive reduction from what I can recall. So there is something there to answer your question, suggesting that there is some resolution, but I don't think it's has been fully answered at this point. Perhaps in depression we can do that. Certainly we cannot answer this question in schizophrenia because there is no increased signal to measure and reduce. Did I answer your question?
Sylvia, are you still there? Are you muted, perhaps? Yeah, please, go ahead. Sylvia, ask another question. Oh, I'll ask you another question. Yes, I can do that. What is the TSPO tracer behavior in the white matter? Is there unspecific binding, or could it be used to measure white matter inflammation?
We tried. There is a paper by Yvonne Surijan where she spent a long time trying to do this. And so we measured TSP. It's a paper measuring TSPO in white matter. The signal is very low as compared to what we see in gray matter. The kinetics are different, so it's very hard to quantify. You can enjoy the afternoon looking at the million experiments that she's done together with Pablo to be able to quantify white matter with FEPPA. This is an ongoing struggle in PET. The kinetics of white matter are different than the kinetics in gray matter, and so it requires a lot of thought on how to quantify TSPO, but it can be done. We've done it, and we should do more of those studies going forward. We have the data, we just have to do it.
Great. If there are there any other questions? Okay, well, thank you so much, Romina. That was a wonderful talk. I don't know if any students want to stick around and ask questions one-on-one with Romina. I think you agreed to stick around for a little bit, haven't you? Or would you be willing to?
I'm happy if I don't know what
so
time.
If anyone wants to, I don't know if there will be anyone, but if there's anyone who would like to ask some questions privately, feel free. So other than that, thank you very much and have a wonderful afternoon.